Hello students, today we are going to discuss about the next part of the topic sexual reproduction in flowering plants. Already we have discussed that the reproductive part of the plant is called as flower, and the flower contains 4 holes. 2 are essential, taking part in reproduction, and 2 are non-essential part. We have also learned about the parts of this reproductive part. The stamen is the male reproductive part, which is made up of anther and filament, and carpel or pistil is the female reproductive part, which is made of stigma, style and ovary. We have also learned about the male and female gamete. The male gametes are produced only during the process of fertilization. They are present inside the pollen grains. Pollen grains, they contain they carry the male gamete. Whereas the female gametes, they are present inside the ovary. The ovary contains ovule. Inside the ovule, the embryo sac is present, and inside the embryo sac, the egg is present. Not only the egg, but some other egg, like female gametes or female cells are also present, which also take part in the process of reproduction in flowering plants. Now, in most of the organisms, you will see when the fertilization is internal, the male gametes need to get transferred near to the female gametes. Female gametes are normally, in most of the cases, they are non-motile, whereas male gametes are motile, or they are moved or they are transferred from their position to another position near to the female gamete so that they can fuse. In plants, the process of transfer of the pollen grains, not exactly the male gametes, but in the form of pollen grains, from the anther to the stigma is known as pollination. Pollination is the process of transfer of pollen grains from anther to stigma in flowers. Pollination can take place in 2 ways. There are 2 main ways by which pollination can take place. First one we are talking about is self-pollination. It is the pollination taking place within the same plant. Okay, within the same plant, either within the same flower or within 2 different flowers of the same plant. If the plant is same, in the same plant, the flowers are used for pollination. If the pollen anthers of pollen grains from anther to stigma are transferred within the same flower, we call it as autogamy. If the anther of one flower is transferred to the stigma of another flower present on the same plant, we call it as geitonogamy. Both are under self-pollination. Autogamy takes place very easily on its own because this anther and stigma, they lie close to each other so that pollen grains can fall from anther to stigma. They are arranged in such a way that the anther and stigma comes in close contact and pollination takes place. Whereas for other part of the self-pollination, that is geitonogamy, when the flowers are different, there are some other ways by which pollination takes place. Before going to that, we also discuss about cross-pollination. It is a type of pollination that takes place between 2 different plants. Flowers are present in 2 different plants which should belong to the same species. Okay, pollination always takes place between 2 same species plants or 2 same species flowers. It is mandatory. So when pollen grains are transferred from the stigma from the anther of one flower present in one plant or present in a male plant to the stigma of the female flower present in the female plant belonging to the same species, we call it as cross-pollination, or another word we use, that is genogamy. Okay, so these are the ways by which pollination can take place. Now, as we already said, autogamy can take place on its own because they are very close to each other, so very easily it can take place. But for geitonogamy and genogamy, there are some other agents needed. Some other external factors are needed which will help in this transfer of these pollen grains. Those external factors are also known as pollinating agents. There are different types of pollinating agents, and based on the name of the type of the pollinating agent, we classify pollination into different types. For example, if wind is the pollinator or wind is the pollinating agent, we call this type of pollination as anemophily. If water is the pollinator or pollinating agent, we call it as hydrophily. This anemophily and hydrophily means by wind and water, these are the non-living or abiotic factors or abiotic abiotic agents of pollination, whereas others are biotic agents of pollination. What are the other agents or biotic agents? Insects, we call it as entomophily. Birds, we call it as ornithophily. Bats, chiropterophily. Animals, zoophily. There will be many other names, like suppose we can call for snail. If the snails are involved, then we call it as myrmecophily. So many other names can be used, but the most common ways of pollination are wind, water and insect. Wind, water and insect are the most common substances or common agents for pollination, whereas insect is the most common. Okay, and it is also seen that the flowers and the pollen grains are modified according to the type of pollinating agent. For example, if the flower has to be pollinated by wind, the pollen grains will be very light. Okay, anther will be exposed, pollen grains will be very light, it will be hairy, so that it can fly with air. If it is water pollinated, then they will be covered with a you can say a protective layer, so that they do not get damaged by the water, and they will be light so that they can float in water. Insect pollinated flower has some very interesting characteristics. The flowers are by seeing the flower only we can understand that this particular flower will be insect pollinated. You know how? The flowers will be very attractive looking, very brightly colored. This bright coloration, which is normally present in the petal part of the flower, are actually due for insect attraction, attracting the insect for pollination. If insect doesn't come, pollination will not take place. And why will the insect come? Only when they get attracted. So either they will be very brightly colored, or they have a very strong fragrance. Okay, and they always contain nectar, because insect come to the flower in search of nectar. So they will try to eat the nectar, they will have the pollen grains attached to their legs or wings, and when they go and sit in another flower for again search of nectar, then this pollen grains get transferred. So this is the way in case of insects. Birds, bats, animals, all these things also take part in the process of pollination in the same or the other way. Okay, so normally insect pollinated flowers, they are very brightly colored, they have good fragrance, they have nectars, and the pollen grains are sticky in nature. They stick to the legs of the insect. You know, for you have seen this night blooming flowers, they are normally white. You know why white? Most of the night blooming flowers are white. If you try to recall, jasmine, rajnigandha, night queen, all are this white, they are white in color. You know why? Because at that time, this insects will not be able to see the color. So there is no use of the color. Instead, they have a very strong fragrance. If you have noticed, or if you can recall now, all these night blooming flowers have a very strong fragrance. Why? To attract the insects. No use of color. Instead, they have the fragrance. Okay, students. Now we know about the process of pollination. We will come to the next step of the reproduction, that is fertilization. Fertilization means fusion of male and female gamete. When we study reproduction, there are some important steps. Number 1 is formation of gamete, number 2 is transfer of gamete, and number 3 is fusion of gamete. We have already discussed about formation and transfer of gamete. Now we are talking about fusion of gametes. The gametes, where are they present? The female gamete inside the ovary, the male gamete in the pollen grain. As pollination has taken place, all are close to each other. Now, how this process of fertilization will take place? I have written in points. You see the points. Number 1 point. The pollen grains will fall on the stigma of the flower. Number 1. Pollen grains will fall on the stigma of the flower. Number 2. This pollen grains will now start absorbing the water and nutrients from the stigma. Okay? And they will germinate. When they will get absorb this water and nutrients, they will swell up and they will germinate. Germinating means they will form the pollen tube. How this pollen tube is formed? What is this pollen tube formed? As we have already discussed beforehand, the structure of pollen grain. A pollen grain, it consists of an outer layer which is rough and an inner smooth layer. Okay, the smooth layer is known as intine, whereas the outer layer is known as exine. There are some points at which this exine is absent and through this, these points are called as germ pores. Why this exine is absent at this point, you know? Because exine is so thick that it is impenetrable. Nothing can penetrate it. So there is no way by which this intine or the cells inside present inside this exine can come out. So for this convenience of this process of pollination, fertilization, these germ pores are present. These germ pores will help the pollen grain to absorb these nutrients, and through the germ pores, the pollen tube will come out. What happens actually is during this pollen tube formation, inside the pollen grain, in most of the cells, I have already told, in most of the cell, like pollen grains, there are two cell stage. Okay, one called as vegetative cell, another is called a generative cell. During germination, the generative cell stays inside, whereas this vegetative cell form the pollen tube. The vegetative cell's function is to form the pollen tube, and once this vegetative cell form the pollen tube, the generative cell's nucleus will come here. Okay, this generative cell's nucleus will ultimately divide into two to form the two male gametes. Okay, this happens in about 60% of the plants. Then rest 40% of the plants, what happens in them? The pollen grain is already in a 3-cell stage. 3-cell stage means one vegetative cell and 2 male gametes. The generative cell is already divided into forming into 2 male gametes. So one vegetative cell for forming the pollen tube and 2 male gametes, which will ultimately come out through the pollen tube once pollen tube is formed. Okay, so this is the process of formation of pollen tube and entering of this nucleus, generative cell nucleus, or the 2 male gametes in the pollen tube is known as germination. Now what happens after this? The pollen tube will continue to grow. Okay, it will continue to grow towards the ovary. You can see the diagram here I have drawn. These are the many pollen tube, many pollen grains are fallen on the stigma. Only if they are species specific, they will be identified by the stigma. Some receptors are there, some identifying factors are there. So when they match, the pollen tubes are produced, the pollen grain germinate. Then not all, but some of this pollen tube, they will enlarge. They will enlarge and ultimately they will reach the ovary. They will keep on growing until and unless they reach the ovary. Okay, so you can see in the diagram, they will reach the ovary. Then you can see the next part. What happens afterwards? When this pollen tube will reach near to the ovary, the tip of the pollen tube will fuse to the wall of the ovule. Now you can see here in the diagram, there are two ends. I have already marked. This particular end of the ovule is known as chalaza, whereas this end is known as micropyle. In most of the cases, the pollen tube always enters through micropyle, but sometimes it enters through chalaza by inverting these integuments, or sometimes through these integuments also. Rarely, but it happens some of the cases. So most of the cases we are drawing through this. You can see this one is through this micropylar end. The pollen tube has entered. The tip of the pollen tube will fuse with this embryo sac wall inside the ovule, and the pollen tube will release the male gametes inside the embryo sac. Okay, the pollen tube will release the male gametes into the embryo sac. How many male gametes are there? 2 male gametes are there, because either the 2 male gametes have entered the pollen tube, or the generative cell nucleus, it has already divided into 2 male gametes. The male gametes are haploid. Okay, remember the male gametes are always haploid. Now once entered inside this embryo sac, what happens? You see, number 1, the first male gamete, which is haploid, fuses with the egg, which is also haploid. Okay, and produces a zygote, and plus and the zygote will be twice-n or diploid. This fusion is known as syngamy. Okay, so fusion of the first male gamete and the egg produces a zygote which is diploid in nature, and it will be the step will be called as syngamy. The second male gamete. Okay, now the second male gamete, what it happens to this? The second male gamete, it fuses with the central cell or the polar nuclei, which is already twice-n, which is already diploid. So 1n plus 1 twice-n, it will result in formation of a primary endosperm nucleus, which is triploid or trice-n in nature. We also call it as PEN, PEN. In short, primary endosperm nucleus, PEN, which is trice-n or triploid in nature. This particular step of fusion of N plus twice N is known as triple fusion. Why triple fusion? Because 3 N's have fused to form a triploid cell. Understood? So this is triple fusion. And now here you can see the fusion is taking place twice. This is a one fusion, this is another fusion. Okay? So as in the process of sexual reproduction in flowering plants, the fusion of male and female cells occurs twice. We call the whole fertilization process as double fertilization. Okay, the whole double process of fertilization is known as double fertilization. So when you are asked to explain the process of double fertilization, you have to explain from the beginning, immediately after falling the pollen grains on the stigma. After pollination you have to start, and you have to complete with the diagram up to this point. If question is asked why pollen grains, or sorry, why this fertilization in angiosperm is called as double fertilization, you can simply write it is called as double fertilization as fusion of male and female cells occurs twice. First male gamete and egg to form zygote, and the second male gamete and polar nuclei to form the primary endosperm nucleus. This should be your answer. The earlier portion description is not required. If question comes in what is triple fusion, then only this part, only this portion you need to answer. Diagram is very, very important. This is a very important diagram. It comes a lot with proper labeling. So I want all of you to practice the diagram as much as you can so that you don't miss any part. You need to show the proper diagram. If you are asked to show the double fertilization, you need to show this, not a flower. You don't have to show the flower. If you are asked to draw a flower, it's a different question. If you are asked to show the double fertilization, you need to show only this part. The sepals, petals and all other things are not required. Okay? Let's now talk about the post-fertilization events. Post-fertilization events means what happens after fertilization is over. The various parts of the flower, they have their own fate after fertilization. The sepals, petals and stamen, which do not have any role during the process of fertilization also. After pollination, even the stamen's role is also over. So the sepals, petals and stamen, they wither off. Number 2, the style and stigma also degenerate. Only ovary is involved. The ovary develops into fruits. Ovule forms the seed. While the integuments covering the ovule, they form the seed coat. The zygote forms the embryo. The zygote which is formed inside the ovule, now it is developed to form the embryo. So we know embryo is present inside the seed, seed remains inside the fruit. Like earlier, the zygote is inside the ovule, ovule is inside the ovary, now embryo is inside the seed, seed is inside the fruit. Now the other part, another part which is formed as a result of fertilization, that is pre-EN, primary endosperm nucleus. The PEN forms endosperm which provide nourishment to the developing embryo. Okay, during the process of development, that nourishment is provided by PEN. It is seen in some of the seeds, the endosperm is completely used up. No endosperm is left after fruit is formed or in the fruit, because the developing embryo has used up all the endosperm. So we call those fruits as non-endospermic seeds. Okay, all those seeds, non-endospermic seeds or non-albuminous seeds. But in some other cases, it is seen that endosperm is left up. Most of the endosperm, or the handful of amount of endosperm is left out, and it becomes the part of the fruit. It is called as endospermic seed or the albuminous seed. You know, there are many examples are there. It is seen that normally you can say different plants can be divided as monocots and dicots, as I think you have already studied about that in earlier classes, in class 9. Monocot plants, where the embryo, when we see the seed or the embryo, seed is having a single cotyledon. Dicot, the seeds are having 2 cotyledons. It is seen that in case of a dicot embryo, the seeds are mostly non-endospermic. Seeds are normally mostly non-endospermic, whereas in case of monocot embryos, the seeds are endospermic. Exceptions are there, definitely exceptions are there, but sometimes most of the cases it happens like that. You have seen coconut. You've seen the water of coconut. That is liquid endosperm. Okay. So we can say ovary form the fruits, ovule form the seeds, zygote forms the embryo, PEN forms the endosperm, which is used for nourishment of embryo. Sometimes it is left, sometimes it is completely used up. Some exceptions are there in these points also. Normally ovary develops the fruits. The fruits which are developed directly from ovary are known as true fruits. But sometimes instead of ovary, some other part of the flower also produces the fruit. For example, apple. In apple, the thalamus part, thalamus I have already explained. Thalamus or now it is also called as receptacle. The thalamus produces the fruit. Apple is formed from the thalamus. So it is known as a false fruit. Okay. Here we are talking about the structure of a dicot seed. In your syllabus, the structure of a dicot seed is given along with a practical. I will show the practical in another class. From whom I will show, you try on your own. Let's today see what are the structures of a dicot seed. A dicot seed, the word dicot means dicotyledonous. Means the seed contains two cotyledons. What are cotyledons? These are the seed leaves. The first seed leaves that are formed from the seed. We call it a seed leaf. When you sow a seed, the seed, it will germinate and it will form the cotyledons. Those are the first leaves. They form the first leaves. So the cotyledons are 2 in number in dicot seed. Okay, so I am drawing an example of a dicot seed here. Example, you can try with gram, beans, all these things. So suppose this is a dicot seed. You can see what I am dividing. The seed has two main parts, seed coat and embryo. So here the outer layer is the seed coat and this one is, inner portion is the embryo. Okay, the embryo again has two portions, cotyledon and embryo exists. So these two, inside the seed coat, these two you can see, these two are the cotyledons. The two cotyledons are covered with the seed coat. And inside this two cotyledon, the embryo remains. The embryo exists actually, the various parts it forms it exists, so we call it embryo exists. What are the various parts? When it will develop, when it will germinate, we will show two parts. One part goes up, another part comes down. This part going up is called as a plumule. Okay, it is the future shoot or stem of the plant. The part going down is the radicle. Okay, I've written here radicle. It is the future root of the plant. Okay, this is the point of attachment. The point at which the cotyledons are joined to the embryo axis is known as the point of attachment. Upper portion is plumule, lower portion is radicle. The area between, or the portion between plumule and point of attachment is known as epicotyle, and the area between point of attachment and radicle is known as hypocotyle. Okay, so this is the structure of a dicot seed. You can try on your own also at home. You take some seed, then let it germinate, soak it in water for 1 day, take it out, let it germinate, let the things come out, it will germinate. So you can see the parts. Then some of the seed you sow in on like soil, you will see that the two seed leaves are formed from the first, like these two cotyledons. First two seed leaves of the plant are these two cotyledons. Okay, thank you.